proceed further, can anybody let me know what is the genetic material in all the living organisms? Are you aware of the term genetic material? Do you know what is the meaning of genetic material and what are the properties of genetic material? Any idea? Maybe if you remember your household things, you can recollect that. Many times what we will be doing, what we will be hearing is maybe when you visit your grandparents, they'll say he's a replica of your uncle or she's a replica of your aunt. Have you ever heard of that? So from that, can you guess what are the properties of genetic material? Okay, you must be knowing any, huh? So let us go directly into that. First of all, what are the properties of genetic material? There are basically four properties of genetic material. The first property of genetic material is it should store the information and it should transmit the information as and when the cell requires. Is that clear? So what is the first property of genetic material? Genetic material should store the information and it should transfer the information to the cell as and when there is a requirement. Is that clear? That is the first property. We are not going into the details of how it is or why do we call it as genetic material. Maybe if the time permits, at the end of the class we will look into all those details. So the first property is we have seen. Then what is the second property? The second property is genetic material should be stable both physically as well as chemically. It should be stable both physically as well as chemically. That is the second property. Okay? Why it should be stable? That is one basic question which you can ask yourselves. Can anybody let me know why the genetic material should be stable? When you say that it is genetic material and it is, what is it doing? It is storing the information. It is storing the information. What sort of information is that? It is the genetic information which is required for our cellular processes. So that's why it is very, very important and very, very vital. Okay? If it is not stable physically and chemically, then what will happen? What will happen? It might change. It's something like this. You have valuable ornaments. Where do you keep them? Inside your residence, maybe in a locker you broke it. And you will put a big lock onto that. Why do you do that? Because thieves or it should not be accessible to everyone and it should be stored safely. Because safety is very much essential. Similarly, the genetic material is also very, very important. So that's why it should be stable both physically as well as chemically. So proper care should be taken by the cell. Proper care should be taken by the cell to ensure its safety. Okay? So that's why genetic material should be very safe. And what is the third property? A cell, what will be doing? A cell will be duplicating. What is the main function of a cell? Cell doesn't want to remain alone. So what it will be doing is it will think of or it will undergo duplication. That process is called cell duplication or cell division. So whenever the cell divides into two, or whenever a cell divides into two, then what is the fate of this genetic material? Is genetic material going to be constrained only to a single cell, or is it also going to get replicated? Then the answer is the genetic material should undergo a faithful duplication with minimal errors. What is that? It should undergo duplication or it should duplicate with minimal errors and a copy of the genetic material should be transferred to both the progeny cells. That is the third property. Okay? So, these are the three properties and in addition to that there is one very important property which is the last but not the least one. That is, the genetic material should undergo certain occasional changes. It should not remain always constant. But it should not remain always constant. Because genetic material, if it doesn't undergo changes at any point of time, there won't be any evolution. There won't be any evolution. All of you must be knowing that the life has originated in the form of a single cell. But right now, if you look at the blue whale, which is the largest mammal, how many cells will be there? Maybe billions of cells will be there in that. From a single cell rather changes, occasionally it should undergo certain changes. Those changes, they should not be so drastic, but they should not be so drastic. They should bring a little bit of variation. They should bring a little bit of variation within the normal characters. So these are some of the properties of genetic material. This is in short about genetic material. Okay? Now, the next question that comes to our mind is, how we came to know that DNA is the genetic material? Today we are sitting in a classroom and we are discussing that DNA is the genetic material. But what about the scientists and what about the people long back? What were the thinking of genetic material? Any idea? Before 1940s and before 1900s, do you know what people used to think? Let me cover. See, Mendel did not say even a single word about DNA. He has used the term factors. See, but he has coined the term factors, which is, he doesn't know what is the basic composition of that. But how do we know today that DNA is a genetic material? Anybody? See, Griffith, someone said Griffith. Did Griffith say anything about the DNA? Practically speaking, he said nothing about DNA. See, the list continues. See, there are certain experiments which led to the development of the tablet, which led to the fact that DNA is the genetic material. So what we will be doing is, we will be looking into the experiments which prove that DNA is the genetic material. We will look into certain basic structural details of the DNA. What is aDNA, what is bDNA, what is zDNA, and what are certain special forms of DNA. Are you aware of the term wDNA and hDNA? We'll look a little bit into them. Okay, so first of all, these are the things which we will be covering in today's class. Okay, the first one is the classic experiments which elucidate the structure of DNA. Then we'll look into how Watson and Crick has proposed their double helical structure, and then what are the types of DNA conformations which we have, and last but not the least is a little bit of information about RNA, excluding mRNA. I'm not talking about mRNA, we will be looking about other RNAs. Okay? And one more thing, at any point of time, if you don't agree with me or if you have any doubt, please stop me. Then and there itself. Is that clear? And it will be glad if you answer the question rather than keeping quiet. Okay? Now, first and foremost thing, there are certain points which are useful for you for your examination. Like, they may ask you may get a question. When nucleic acids were discovered for the first time. So what is the answer? It's way back in 1870. Okay? And why did they call them as nucleic acids? They discovered nucleic acids way back in 1870. First of all, what are nucleic acids? Can anybody tell me what are nucleic acids? Think logically. What are nucleic acids? Acids present within the nuclei or within the nucleus. Okay? So when you say that they are acids, they are acidic in nature. That is first point. Second thing, when you say that they are present in the nucleus, you are talking about you are localizing their existence. Where do you find them? You know where do you find them. Okay? So that's why you are calling them as nucleic acids. You are referring to by calling them nucleic acids, you are referring to two points. One is the charge or one is their nature, and the second one is where do you find them. Okay? So these 2 points, they are expressed in this by calling the term nucleic acids. Then, what do they contain? What nucleic acids contain? It's written over there. See, I want an active participation from you. I don't want to talk to students who are mom. So, what does it contain? Basically, it's a weak acid and it is rich in phosphorus. Okay? Then, what else is written over there? If you look at the name, what is them are known to you. Then it is a component of the chromosomes. And last but not the least, what is the last but not the least one? What was written there? It is the carrier of genetic information. Okay? Now, of course, if you go into the timeline, Kadi has discovered that way back in 1969, and then in 1928, the first, before we proceed further, let me stop over here. Yeah. Now, how do we come to know that DNA is the genetic material? There are basically three sets of experiments which prove that DNA is the genetic material. The first set of experiments, they are known as transformation experiments. The second set of experiments, they are called chemical experiments, and the third set of experiments, they are known as blended experiments. Let me repeat this once again. And see, in different books this will be given differently, but the mostly followed version states that there are three sets of experiments which prove that DNA is the genetic material. So what is the first set of experiment? Transformation experiments. Remember that these are called transformation experiments. They were not carried out by a single scientist, but they were carried out by certain groups of scientists. That's the name, transformation experiments. The second one is chemical experiments. Third one is blended experiments. First of all, let us look into transformation experiments. Now, way back in 1928, Griffith, he was experimenting on mice and he was working with a bacterium that causes pneumonia. Okay? That causes pneumonia. When he was working on that, he has isolated different strains, but among them there are two strains which are noteworthy. Okay? One strain is called R strain and the second one is called S strain. Very good. Now, what is the primary difference between R strain and S strain? R stands for rough and S stands for smooth. Then the next question that comes to our mind is, why the S strain is known as smooth strain? What is the reason behind we calling it as S strain R? Why? Griffith called it as S strain. So what is the answer? Why? The S strain is called smooth strain because it has a polysaccharide capsule surrounding the organism. Around the organism, a polysaccharide capsule is present. Okay? So because of the presence of this polysaccharide capsule, smooth-edged colonies are being produced on an agar plate. So you can see there is an agar plate. Can you see that? Okay? On this agar plate, when you grow the organism, if you grow the R type of organisms, the colonies that are developed, they just, you see, they are rough-edged. Whereas that of the S type, their colonies, they are smooth-edged. Hence the name R type and S type. Some people also call them as virulent and virulent strains. Virulent strains are those which cause pneumonia and the virulent strains they do not cause pneumonia. Okay? Then one basic question, of course this is known to all of you. Now, I have a doubt and I want to clear my doubt. My doubt is, why pneumonia is caused in this mice? Why pneumonia is caused? Can anybody answer this question? Or what is responsible for pneumonia? Of course, it is S type of bacteria that are causing pneumonia. Which component of the bacteria is causing the pneumonia? Anybody? What they have found out is, what the scientists have, what he has found out is, the polysaccharide capsule which is surrounding the bacteria is responsible for this virulence. Okay? And our type of bacteria, they lack this polysaccharide capsule, so that's why they are not causing the virulence. So, virulence, it is associated with this polysaccharide capsule, but he doesn't know what is responsible for this polysaccharide capsule appearance in S type of bacteria. And he did certain experiments which have been briefed over here. So, as a part of his experiment, first what he did is, he has taken a group of mice. He has taken a group of mice and he has injected these mice with the S type of bacteria. bacteria, the live S type of bacteria. When you inject the mice with this live S type of bacteria, what will happen? Bacteria, they have the polysaccharide capsule. So because of the polysaccharide capsule, they will be causing pneumonia and will be killing the mice. So the mice die. That's a known fact. Then the next thing is, they have taken R type of bacteria which are alive, and the mice, they have been injected with R type of bacteria. So when you inject live R type of bacteria, are the mice going to die because of the pneumonia? Remember that R type of bacteria, they lack the polysaccharide capsules surrounding them. That's the first point. So when they lack the polysaccharide capsule, can they create virulence? Because polysaccharide capsule is responsible for the virulence, and they lack this polysaccharide capsule. So that's why virulence was not created. So that's why the mice did not die, even though the bacteria were growing in the mice. Then, so this is the first experiment. Then what they did is, they have taken the S type of bacteria which create virulence. They kill the bacteria. When they kill the bacteria, and when they injected this S type of killed with S type bacteria into the mice, what do you explain? Did they die or not? He wanted to know, so that's why he did an experiment, and he has found out that by injecting heat-killed S type of bacteria into the mice did not die. So he came to know that if you heat-kill the S type of bacteria and if you create a cell lysis solution and if you inject it into the cell, the mice did not die. If you look at here, there are two types. One is the bottom one which do not kill the bacteria, and the right bottom one which also do not kill the bacteria. As both of them cannot individually kill the bacteria, what he did is, he has mixed both of them and he has injected them into the mice. And he wanted to know what is going to happen. Okay? So, by mixing both of them and injecting into the mice, he has found out a totally new discovery. What is the discovery? The mice, they died because of pneumonia. And he wondered, if you see, if you take only the S type of bacteria, heat kill them by exposing them to heat, and when you inject them, the mice did not die. Similarly, when you take live R type of cells, if you inject them, then also the mice did not die. But when you mix both live R type of cells and S type, heat-killed S type of cells, then the mice died. And what he did is, from the dead mouse, he has isolated the bacteria and he has grown them on the Berkeley plate. And he has found out the bacteria which were isolated, they belong to the S type or smooth-edged colonies. Okay? So, he is another of the reason, but he has concluded that the R type of bacteria, they were transformed into S type of bacteria. He did not know the exact reason, and he has put a plausible reason as mice must be responsible for the transfer of or for the transformation of R type of bacteria into S type of bacteria. This is what Griffith has concluded. Griffith never stated that DNA is the genetic material. He did not even a single word about DNA. Okay, what I have seen is many students, they were stating that Griffith has concluded that DNA is the genetic material, but in fact, he did not utter even a single word with respect to DNA. Is that clear? Okay, then this occurred way back in 1948, and then for almost 16 years nobody touched this. Then they were wondering what is the chemical nature of transforming things. Then there were two schools of thought. One school of thought, they used to propose that DNA is the genetic material. The second school of thought, they used to propose that proteins, they are ideal candidates for being the genetic material. By people who used to support proteins, we know why. Some people they used to promote proteins, or why they used to say that proteins are ideal candidates for being the genetic material. Are you aware of the reason? if you look at the DNA is only made up of 4 nucleotides, whereas proteins, if you see, they are made up of at least 20 amino acids. Okay, so where do you find a greater variation? Where do you find a greater variation? Is it in case of DNA or is it in case of proteins? Where do you find a greater variation? Proteins are made up of at least 20 different amino acids, and DNA it is made up of only 4 different nucleotides. So you could see a lot of variation in proteins than DNA. This is what people used to presume. So people who were supporting proteins as the genetic material, they were stating that there are at least 20 different amino acids and there are great variations. These great variations, they are responsible because of this great variation in amino acids. This is what people used to presume. Okay? This is the existing belief net. Then after 16 years, every McLeod and Kadi, they repeated this experiment and they almost proved that DNA is the transforming principle. But what is that? But we will see a little while later. First of all, what did this every McLeod and Kadi do? First of all, they wanted to know what is this transforming principle. They wanted to know what is this transforming principle. So further what they did is, they wanted to know whether mice are really essential for the transforming of R bacteria into the S bacteria. So that's what they did basically is, first of all they have taken S type of bacteria, they have subjected it to heat killing, so they took that cell lysate and they have directly added the cell lysate to the R type of bacteria. And they saw whether any R type of bacteria have been transformed to S type of bacteria or not. What did they find out? They found out that transformation is taking place even when only the cell lysate from the S type of cells if it is added to the R type of cells. However, with a very low frequency, something like 1 in 10,000. Okay, 1 in 10,000. So, for transformation to take place, the first thing they have to do is, for the transformation to take place, mice are not essential. Only S type of bacterial cell lysate is sufficient. Okay, that is the first thing. The second thing is, they did a few more experiments. To prove what is the nature of this genetic material. So first of all, they have taken the S type of bacteria, they heat kill that, and they have tested whether it is really converting or transforming R type of bacteria into the S type of bacteria or not. When they came to know that it is transforming, then they proved that they have purified that transforming principle. They have purified the transforming principle, and they did certain experiments. The first thing they did is they analyzed it chemically. That is, they have carried out the chemical analysis, and they have also carried out physical analysis. What do you infer from this physical and chemical analysis? By observing, by looking at the boiling point, you can say what is its closest compound. Okay, that is equivalent to, that has equal boiling point. Okay, similarly, what is its density? Okay, what is the chemical nature of that? When they did that, they came to know that this transforming principle is a nucleic acid that contains deoxyribose as the sugar. This is what they came to know. That is the first one. But it also contained a little bit of proteins, RNA and other things. Okay, then what they did is, they have taken the transforming principle and they wanted to prove that, they wanted to know its nature. Okay, so that's why what they did is, initially the transforming principle was taken, it was treated with the enzymes proteases. Now, what is the function of these proteases? What is the function of proteases? Proteases, they breaking the proteins. Okay? If transforming principles were made up of proteins, then when you treat them with the proteases, then transforming principle should lose its activity. So they have treated the transforming principle with proteases and they have subjected the transformation and they found out that transforming principle did not lose its activity. Okay? Now a question to you. How do you prove that transforming principle is not made up of RNA? Now, when you treat the transforming principle with RNases, is it going to lose its activity or not? If it is going to lose its activity, what do you infer? And if it is not going to lose its activity, what do you infer? What is the conclusion you are going to draw when the transforming principle is treated by RNAs and if it loses its activity? What is the meaning of loses its activity? Anyone? Anybody? That is, normally what do you observe when you add the transforming principle to R type of bacteria, 1 out of every 10,000 type of R type of bacteria were being transformed into S type of bacteria. Okay, that particular property of transformation, it will be lost. Okay, it will be lost. That is the meaning of losing its property. Okay, so from this they came to know that by carrying out all these experiments, they came to know that transforming principle is nothing but DNA. However, they dare not say that transforming principle is DNA. But what did they say? They have stated that nucleic acids, they have a specific role which is yet to be assigned. Okay? Why did they say like that? They could well have stated from these experimental results that DNA is the genetic material. But they did not say so because legal circumstances force them to state that nucleic acids have a specific role, but we don't know what is that role because people who were promoting proteins as the genetic material were high in numbers. Okay, so these were the experiments carried out by Abri, Metia and Kadi. Later, from this they could well have stated the transforming principle is DNA. Then came Erwin Chaga. Who is Erwin Chaga and what is his contribution? Erwin Chaga carried out a set of experiments known as chemical experiments. Before Chaga, what people used to do is they used to estimate the nitrogenous basis ratio within the DNA molecule. Okay? And there used to be people used to have a preformed notion that all the nucleic acids they have equimolar concentrations of 18, thymine, guanine and cytosine. That particular philosophy has been disproved by Chaga. Okay? So Chaga from his experiments he has proved that the ratio of adenine plus thymine or adenine is equal to that of guanine and guanine is equal to that of cytosine. In other words, he has concluded that the molar concentration of purines is equal to the molar concentration of pyrimides and it is not that A is equal to E is equal to G is equal to C. Previously, scientists were presuming that all the 4 nucleotides they used to be equal in their concentrations, which was disproved by Erwin Chaga. Okay? And so when Chaga has published these results, people started stating that DNA may be genetic material and it could be a plausible candidate for being the genetic material. However, unless and otherwise somebody confirms that you cannot state that DNA is the genetic material. And last but not the least, then Pesci and Chase carried out a simple method experiment Hershey and Chase, they were doing a simple experiment. I hope all of you are aware of the term bacteriophages. What are bacteriophages? Bacteriophages are viruses that infect the bacteria. Okay? The known thing is that the bacteriophage will be absorbing to the bacteria. It will create a hole. It will inject its DNA, and prophages will come out by killing the bacteria. This is a known fact. Okay? And Hershey and Chase, they carried out certain experiments to prove the DNA is the genetic material. In other words, they did not start with DNA as the genetic material. They wanted to know what is the genetic material. But what is the reasoning they did, or what is the groundwork they did before they experimented? Can anybody let me know? Why they have carried out their experiments? And how did they prove the DNA is the genetic material? What they did is, a bacteriophage will be absorbing to the bacteria. When a bacteriophage gets absorbed to the bacteria, it will be transferring its genetic material. After its genetic material is transferred, if you shake them vigorously in a kitchen blender, you can separate the bacteriophage from the bacteria. This is what they came to know, and they have applied it in a complicated way. What did they came to know? When a bacterium is absorbed by the